The modern pharmaceutical industry operates on a documented business model that routinely prioritizes market share over patient safety. We saw this clearly when Purdue Pharma aggressively marketed OxyContin as a non-addictive painkiller, directly fueling the opioid crisis to maximize profits. In 2009, Pfizer paid $2.3 billion in the largest healthcare fraud settlement in history at the time. They were caught falsifying safety data and bribing physicians to prescribe drugs for unapproved, dangerous uses. How does an industry with this specific criminogenic history secure global mandates for experimental therapeutics? They bypass the regulatory firewall. A quantitative study of the Department of Health and Human Services found that 54% of CDC personnel leave their public roles to take lucrative positions directly within the private industries they were meant to regulate. This financial overlap extends globally. In the United Kingdom, the Medicines and Healthcare Products Regulatory Agency receives 86% of its operational funding straight from the pharmaceutical sector. The institutional firewall between the regulator and the regulated has collapsed. Health agencies now function as corporate partners, creating an iron cage that protects industry revenue rather than acting as independent scientific watchdogs. When independent researchers finally accessed and reanalyzed the original Pfizer and Moderna clinical trial datasets, they applied the Brighton collaboration's strict definitions for adverse events to see exactly what the manufacturers missed. This chart shows the absolute risk increase of suffering a serious adverse event following vaccination. In the original healthy trial cohorts, the data reveals a risk of 1 in 800. These were not mild reactions. That 1 in 800 figure is driven entirely by severe complications, specifically coagulation disorders and acute cardiovascular injuries requiring hospitalization. To understand the risk calculus, you have to look at the number needed to vaccinate. For individuals in their 30s, it takes over 300,000 doses to prevent a single severe hospitalization. Pushing a 1 in 800 risk onto millions of healthy people guarantees a disproportionate accrual of injury. This mathematical anomaly mapped directly onto the real world. Actuarial data from 2022 and 2023 showed sustained non-COVID excess mortality spanning the globe. The Australian state of Queensland provides a unique control variable. In 2021, the state maintained virtually zero community transmission of the virus. Yet, precisely as the vaccine rollout accelerated, older demographics experienced a sharp, sudden spike in all-cause mortality. The raw mathematics prove the medical intervention caused significantly more severe harm than it prevented in healthy populations, a signal that captured regulatory managers chose to ignore. Morticians and vascular surgeons soon began pulling physical evidence of this cardiovascular failure directly from patients. They discovered structures clinically termed anomalous intravascular casts. A normal blood clot is a fragile cellular net made mostly of red blood cells. When the body needs to clear it, it easily dissolves. Surgeons are extracting highly abnormal casts. They are pale white, rubber elastic, stretching up to 25 centimeters, and conforming perfectly to branching human blood vessels. Mass spectrometry testing reveals these structures have a highly skewed ratio of fibrinogen proteins. More importantly, they contain almost zero plasminogen, the specific enzyme the human body requires to break down and dissolve clots. Because these host-manufactured casts lack the biological machinery to self-resolve, they act as permanent physical blockages. They anchor into major pathways, suffocating tissue, and triggering sudden cardiovascular failure. Oncologists are tracking a second, equally alarming biological anomaly: early-onset malignancies that rapidly accelerate to multi-organ metastases in a matter of weeks, colloquially termed turbocancers. To understand why this is happening, look at a tumor suppressor protein called p53. It acts as the ultimate guardian of the human genome. When cells mutate, p53 hunts them down and forces them into programmed cell death before a tumor can form. Independent laboratory studies show that the transfection of the synthetic spike protein profoundly inhibits p53 activation. It paralyzes the cell's natural defense machinery. At the same time, the lipid nanoparticle platform triggers a surge of inflammatory cytokines. This forces rapid blood vessel growth, pumping fuel directly into the mutated cells. The mRNA platform strips the cell of its primary tumor suppressor, while flooding the local environment with inflammatory fuel. Dormant or newly mutating cancers are suddenly free to hyperprogress and metastasize in weeks rather than years. Repeated dosing with this lipid nanoparticle technology induces a third biological consequence: systemic immune tolerance. A healthy immune response relies on highly inflammatory antibodies. They aggressively hunt down viral threats and signal the immune system to clear the infection. Independent studies confirm that after booster doses, the immune system switches its strategy, shifting to mass-produce an anti-inflammatory molecule called immunoglobulin G4, or IgG4. IgG4 is a tolerogenic antibody. It binds to a threat, but it actively fails to signal the immune system's effector functions to destroy it. The immune system is chemically instructed to stand down. This class switch grants continuously circulating spike proteins and aggressively mutating cancer cells a biological free pass to evade systemic immune surveillance entirely. The lethal vascular casts, the paralyzed tumor suppressors, and the chemically induced immune tolerance all trace back to the macro-institutional failure of the regulatory state. The decision to maintain the rollout, despite a 1 in 800 severe injury rate, points to a captured managerial class, prioritizing corporate profitability over its regulatory duties. The foundational principle of the scientific method, the rigorous skeptical testing of physical reality, and the falsification of hypotheses, has been discarded. We are now operating in a closed system of settled science dogma. Political correctness and institutional mandates silence true subject matter experts, while physical evidence of harm is actively suppressed. We must completely decouple pharmaceutical industry funding from public health regulation. To stop the ongoing iatrogenic harm, we have to rescue science from its managers and return medicine to the empirical data.